Professor Loganathan. I think you all have heard of this really powerful technology that got the Nobel Prize in a very short time, called CRISPR, which Dr. Loganathan is going to talk about. So it's a really great pleasure, and thank you very much, Dr. Loganathan, for taking your time to give your presentation. So Dr. Loganathan is an assistant professor in the Department of Otolaryngology, head and neck surgery, and he's also a junior scientist at the MUHC. He is affiliated with experimental surgery, the Goodman Center, and our department. So he received his Ph.D. from the University of Alberta, and in the Department of Biochemistry. So he was investigating the role of membrane proteins in corneal eye diseases. And then in 2015, he joined the lab of Dr. Daniel Schramek as a postdoc at the University of Toronto, where he studied head and neck cancer using CRISPR-based mouse models. So it's a great pleasure to hear his talk, and the title is Applying CRISPR-based Technologies in Mouse Models of Cancer. Dr. Loganathan, please.
Thank you, Dr. Molly. Thank you for giving me this wonderful opportunity to present my research before the members of the Department of Experimental Surgery. Actually, I presented a short 10 minutes version of this talk at the Cancer Symposium last week. So if any of you are there, this talk will be more detailed and with lots of explanations. So with that, okay. So well, I guess you all will agree with me when I say we are living in the midst of genetic technology revolution. What I mean by that is, after Human Genome Project, sequencing technology grew so fast, and now we have entered the era of omics, right? Like genomics, proteomics, things like that. And through these technology, these powerful sequencing technologies, we are generating vast amounts of data. And if you see the slide, and we have generated multiple tools, like, everybody heard about whole genome sequencing, but then after that RNA-seq, single-cell RNA-seq, and then the interaction between chromatin and the proteins, and like so many things. But there are a lot of things, so I just put this flow chart, and you can appreciate all these different methodologies. And this amount of methodologies giving us a never-before sneak peek into the chromatin DNA world, which is great. We are learning a lot. However, we have one new challenge brewing before us. How do we validate and test the functional aspect of whatever we are learning from this vast amount of data? Or even sometimes, how are we going to interpret this data? Well, there are so much of different technology, analysis or assays or experiments available. But one thing I wanted to talk today about is, how do we apply like a genome editing technology, CRISPR, to make sense of this, particularly in cancer. Well, people have used CRISPR so much in vitro, but the biggest challenge is using this in how to use this in mouse models. So today I'm going to talk about 3 different examples of how we take a large set of data and applied CRISPR and identified mechanisms like real drivers genes, and also how we are also developing better systems. So first, I'm going to give an example of whole genome sequencing, where there are thousands of mutations identified in a particular cancer type. And from that, how we can identify the real driver mutations. And second example I'm going to give is how to take an RNA-seq data and then identify either downstream targets or druggable targets using mouse models. For today's talk, just because of time, I'm just going to talk about downstream targets. And also, I will give some work in progress in the lab here at MUHC. And then I will finish off with how we are developing a new system where we can do multi-gene knockout that can be useful for interpreting other omic techniques in a broad sense. Okay. So here is a chart I have where it lists all different types of cancer on x-axis, and then the mutations per tumor on the y-axis. And you could see, for example, the colorectal and lung cancers have more than 100 mutations per tumor in a patient, and all the way down to like a few mutations in pediatric cancers. Yes, we can understand why pediatric cancers have less mutations, but then if you look at all these cancers, there is minimum of 50 mutations per tumor in every tumor. So how are we going to like take all this amount of mutations, and if you add multiple patients, the number of mutations present in each cancer comes to close to thousands or even more than that, right? So for today's talk, and my research area of expertise is in head and neck cancer, so I'm gonna take head and neck cancer as an example and how we did it, but this can be applied across most of the cancers as long as mouse model exists. Okay, just a sneak peek into head and neck cancer. It is the malignancy of the oral tongue, nasal cavity, and the pharynx regions of the head and neck region in humans. And it is the 6th most cancer and 8th in causing death. And 5-year survival is about only 50%. And there is more than 1 million cases every year now. And then the risk factors include tobacco, alcohol consumption, and HPV or EBV infections, and Fanconi anemia. So just like I was telling before, if you take all the mutations in head and neck and tabulate them on x-axis and then put the frequency on the y-axis, you could see a few genes that have a high frequency, which you might have heard from all the cancers like P53, PI3 kinase, CDKN2A, and stuff like that. But then after a few of those genes, suddenly it starts tapering off, and you could see there is a huge long, like this long tail of mutations, which occur like around 5 to 10% or even less than that in the patients. So the question is, not all patients carrying, like say, take P53, there is 70% of head and neck patients carry P53 mutation, but not all of them have a similar prognosis. So the question becomes then, is there any real drivers in this mutations, in this long tail of mutations that has been identified the whole genome sequencing? And will that has any functional aspect to it, right? Or the, how do we functionally test and find these real drivers? Or this long tail of mutations just because of stochasticity of cancer evolution? Or is it highly penetrant but simply affect rarely mutated genes, and that's why we are seeing it in a very less frequency? Or do they converge on a same cellular signaling pathway? These are the questions, possibilities from this long tail of mutations in cancer. But then finding the real drivers is just like finding needle in a haystack. So when my postdoc supervisor explained me this project, literally, you won't believe me, I was just feeling like that. Okay, so how do we do that? The gold standard in the field is obviously to use mouse models for cancer studies. But there is a big drawback, which is that you cannot screen more than 2 to 3 genes at the same time. And even to get to the 3 genes level, you are crossing the mice for several months and up to a year. I mean, it's time consuming, labor intensive, and to screen hundreds of genes, it will take several decades. So how are we going to do this? And that's the time CRISPR was just new into the field, maybe about a year old. And then we come up with this scheme of testing for the real drivers. So what we did do is, I was showing you previous slides, the human HNSCC sequencing data. We take this long tail of genes, synthesize oligos, which is 20 base pair sgRNA from that we use to knock out CRISPR. Sorry, we knock out genes using CRISPR technology. So we can synthesize that 20 base pair sgRNA synthetically, and then clone into a CRISPR plasmid, and then package into lentivirus, and then inject into E9.5 embryo, which at that embryonic stage is a single stem cell layer. So then the genetic information from the lentiviruses will be carried on into the adulthood. And when the pup is born, a single pup carries more than 500 knockout clones on its skin and head and neck surface. So every single colored dot is a knockout of one gene, right? So that's how it works. And then if there is a real driver among those genes, it will grow out as a tumor, and then you can sequence it and you can identify the tumor suppressor. So this is the overall scheme of what we did and how we did it. We took 4 lines, oncogenic mouse lines, crossed them with LSL-Cas9 GFP, and then inject the lentivirus at E9.5, which carries an enzyme called Cre recombinase, and then either the sgRNA against 500 head and neck library or a control library, which is a non-targeting sgRNA. So what I mean by here LSL, so all these mouse has a lock-step block cassette, which means they do not express the oncogene or the Cas9 GFP normally. Whenever we deliver the Cre recombinase in the lentivirus into one single cell, that particular cell, the Cre recombinase removes the stop codon and turns on the expression of oncogene and also turns on the expression of Cas9 GFP. And this Cas9 GFP together with the sgRNA from head and neck library will start knocking out the genes. So we did multiple of these injections, and then finally, this is what we got. So this is a Kaplan. This is a Kaplan-Meier plot where you have time in x-axis and then tumor-free survival on y-axis. All the 4 mouse lines injected with the control library did not form any tumors until at least a year of age. But if you look at the mouse that were injected with head and neck library, be it PIK3CA oncogenic mouse or HRAS or P53, they formed rapid and aggressive tumor suppressors. And from this single Kaplan-Meier survival plot, you are almost looking at the effect of almost 2,500, more than 2,500 genetic interactions. So we collected more than about 200 tumors, sent it for sequencing, and then what we found was ADAM10, RIPK4, Ajuba were the topmost tumor suppressors. And then there were another 10 to 15 new tumor suppressors we identified. In aspect of time and not to overburden with so much of data, we did a thorough investigation of all these 3 top tumor suppressors and employed multitude of techniques and found that in addition to these 3 top tumor suppressors, some of the tumor suppressors we identified all converges on Notch pathway. So let me walk you through it. As soon as the external ligand signals, Notch is cleaved by ADAM10, which then signals the gamma secretase to cleave the internal domain of the Notch, which then translocates to the nucleus and turns on the transcription of downstream genes. While this is happening, NUM is a negative regulator of Notch, which comes and binds to Notch and degrades it, right? But then we found out that Ajuba, which is a new Notch regulator that we discovered, and one of our top tumor suppressor hits, actually sequesters NUM and prevent it from degrading Notch. And then RIPK4, one of the other top tumor suppressor hit, is actually a target gene of Notch transcription. And then some of these ADAM10 and Ajuba are also transcription target of Notch, thereby implicating there is a positive feedback loop driving this Notch pathway. So what does this mean for patients, relevance to human patients? When we went back into the TCGA data and then plotted all these mutations that we find, the genes that we identified as tumor suppressors and the real driver genes, we found that most of them are mutually exclusive. And if you add them all, it added to almost 70% of the patients carrying the mutations in these genes, making HNSCC a Notch-driven disease, which then brings us to our initial hypothesis. Out of 3 possibilities, the real drivers in the long tail mutations are converging on a Notch pathway. This is in head and neck cancer, but we also found something similar that in breast cancer in Daniel's lab, and that paper was just out in Cancer Discovery this week. Okay, moving on. So what next I wanted to talk about is how can we take the RNA-seq data and even use that vast amount of gene profiling data and identify downstream targets using mouse models and CRISPR. For this, I'm going to take RIPK4 as an example, one of the top tumor suppressors I showed you in the previous story. So before doing that, we wanted to make sure, is there a possibility of treating this disease, right? That's the first question we wanted to ask. So we took the RIPK4 mouse. As you can see, if you look at the wild type RIPK4 and the heterozygous RIPK4, if you knock out the if you look at those mouse in the presence of oncogenic PIK3CA, they do not form tumors. But if you knock out RIPK4 in the presence of oncogenic PIK3CA, which is indicated here by the red line, you can see the mouse form rapid and aggressive tumors within 8 weeks. So in the same mouse, in the same every single tumor, we also overexpressed either a wild type RIPK4 that is indicated by green dotted lines, or a kinase dead RIPK4. As you can see, the kinase dead RIPK4 almost resembles the knockout RIPK4 phenotype, versus when you express the wild type RIPK4, the tumors never formed, which indicates that you can actually rescue this tumor formation if you can treat them later. So encouraged by this, we wanted to next study the downstream mechanism of this RIPK4 tumors. So if you knock out RIPK4 in the presence of PIK3CA oncogenic mutation, you get the tumor very rapidly. From getting from here to here, on the way, we know that there will be some genes that will be upregulated and some genes that will be downregulated. So we wanted to employ the RNA-seq technology to identify the genes that are up and down regulated. So how we did that? We took the oncogenic PIK3CA mouse, crossed it to Cas9, and injected with RIPK4 or scrambled Cre, and then we isolated the cells from P4 pups to identify the tumor initiation events, and then also the tumors to identify the late stage events. And when we graph the upregulated and downregulated genes from both, we get a list of genes that are differentially regulated in both. So now we wanted to ask the next question: which of these downregulated genes is the actual effector of the RIPK4? So to identify that, we went back into the same mouse that is oncogenic PIK3CA and LSL Cas9 GFP, but then this time we generated a CRISPR library against the top 100 downregulated genes along with Cre recombinase, just like I was explaining you before, the model where the Cre recombinase will turn on Cas9 GFP and PIK3CA, in addition to knocking out each gene from this RNA-seq list in each clone, each cell on the skin of the mice. And then we monitor for tumor growth. Remember, the RIPK4 is remaining as a wild type in this one. So if there is a tumor formation, that's solely because of the gene knockout that's downstream of RIPK4. So when we performed this experiment, we did got tumors in the mouse that were injected with the CRISPR library against the downregulated genes. And when we sequenced this tumors, we found a gene named E11L4 is the major tumor suppressor downstream of RIPK4. So we wanted to validate this data. So we went ahead into the same mouse, and then we injected a new sgRNA against E11L4, and they formed tumors as rapidly as the RIPK4 knockouts. So they are almost phenocopying the RIPK4, even though it's downstream of the RIPK4. So this clearly tells that E11L4 is the downstream target of RIPK4. But then, interestingly, around this time, there was a paper published in Nature that said RIPK4 phosphorylates IRF6, and this signaling axis actually is important for skin differentiation and barrier function, which was very quite interesting. Why? Because in other 10 new tumor suppressors we identified in the CRISPR screen, IRF6 was also among them. So we then started investigating along the lines of the signaling axis. And then again, I'm gonna not show all the details about it, but I'm gonna show you the final conclusion that what we got or what the data showed. We all know, I showed you already RIPK4's downstream target is E-level4 through the CRISPR screening in mouse models. And then we already saw that RIPK4 phosphorylates IRF6. So our hypothesis was that RIPK4 might be phosphorylating IRF6, which then goes on and regulates the levels of E-level4, which could be forming an axis of tumor suppression in addition to Notch. Why? Because as I have shown before, RIPK4 is a target of Notch transcription. So therefore, Notch regulates RIPK4, which then goes on and phosphorylates IRF6, and then IRF6 goes and regulates E-level4. This signaling axis might be an important axis of tumor suppression in head and neck cancer. We don't want to stop it here. We want to dig a little bit deeper about E-level4. So what is E-level4? E-level4 is the a gene that's responsible for synthesizing very long chain fatty acids. And it's a family of about 8 genes, and 1, 2, 3, 5, and 7. They all play a role in elongating carbon 20 to 30. E-level4 plays a role in elongating the fatty acids from carbon 30 up to carbon 38. So when you look at the E-level4 protein, it has majorly three important motifs: N-glycosylation motif, catalytic histidine motif, and ER retention motif. So we had an question, and also the mutations in this E-level4 gene has been implicated in several neural developmental disorders. And so we had the question, which of these motifs are important for tumor suppression in head and neck cancer, or none of this is important, there is some other function for E-level4? Well, the first step is to first rule out whether any of these motifs are involved in tumor formation. So how we do that? Again, we combined classical biochemistry mutational studies with CRISPR and mouse models, and we wanted to find out what's going on. So you and Advay, the 2 graduate students in my lab, made this plasmid, which actually has an sgRNA against E-level4, which then goes on and knocks out the endogenous E-level4. But then they also express E-level4 with all the different mutations, right? For example, histidine core motif mutation here, and then a recombinase. And then you go back into the PIK3CA-Cas9 mouse, as I was showing before, and what happens is when you inject the lentivirus, just to recap quickly, the Cre recombinase will turn on the oncogenic PIK3CA expression, GFP expression, and Cas9 expression. Oops, sorry. And then sgRNA against endogenous E-level4 will knock out the E-level4 gene. And then the mutated E-level4 will be expressed in the same cell. Oh, crap, sorry. In the same cell, like I was showing, either the N-glycosylation motif or the catalytic histidine motif or the ER retention motif. So now, as we know from previous data, that E-level4 gene knockout will form tumors in 8 weeks. If we overexpress Y-type E-level4, we should rescue the tumor formation. Now the question in between is, which of these mutated E-level4 constructs either rescue the tumor or allow the tumor formation to go through? If it allows the tumor to grow, that means that mutated domain is responsible for tumor suppression. If it rescues the tumor, then that particular domain is not necessary for tumor suppression. So this is an experiment in progress, and currently you and Advay cloned all the different constructs, and we made the virus and we already injected into the mouse. So we are waiting for the results. So the third part of my talk is about developing better systems. Why? Because as these genomic techniques are developing so fast, we need even more complicated modeling, right? Which involves, activating a gene or editing a gene, inducing mutations very quickly, or like if you want to test several point mutations of a gene in a single mouse, you want to have that technology, or even multi-gene knockouts, right? Like you want to create several gene knockout in a single cell. Well, those are all exciting technologies that might come to light one day, or it's already there in vitro, but somebody will adapt it for in vivo. But what we wanted to do is take a first step towards that. So we wanted to create a system in mouse where we can do multi-gene knockout in a single cell. So in 2020, there was this paper in Nature Biotechnology just a year ago, where they were developed this new technology called Chimera, which is basically expressing Cas9 and Cas12 in the same cell, and then driving sgRNA against a against a gene of your choice using Cas9 tracer, and then also use Cas12 guides to target other set of genes. So what happens is Cas12a has inherent cleaving mechanism, so which then if you have the direct repeat sequence, it actually recognizes that and cuts and releases the guide separately. So right now it releases the Cas9 guide, and then you also have a separate Cas12 guide. These two guides can go and do a combinatorial gene editing. So in this paper, they showed that not just one guide against Cas9 and one guide against Cas12a works, but 3 guides against Cas12a also works as efficiently, which means you can technically knock out 4 genes in a single cell at the same time. So this was great. And they showed this in vitro, and the advantage there is that you can take a cell in a dish, overexpress Cas9, select the cells for Cas9 expression, and then again overexpress Cas12a in those cells, select for the cells that are expressing both Cas9 and Cas12, and then you can put this sgRNA and then do the gene editing, which involves multiple rounds of transfection, transduction, whatever method you are using, which is great. But the problem with mouse is we don't have that luxury, right? Like at least with our methodology, if you're going with lentivirus, it has to be one shot and one transduction in the cell, and that's all we have, right? So we are looking to develop or test a technology where we can adapt this to the mouse. So how we do this? This is basically the idea, or the schematics of what we are doing, and we have cloned already some of this, and I will walk you through what is the schematic. So we will take a Cas9 GFP and an LSL TD tomato mouse. They will be normally not expressing Cas9 or GFP, or even tomato, which is red fluorescent protein. So they will be colorless, no green or no red, and no Cas9. We take a plasmid where it expresses Cas12a and then also a Cre recombinase, and together with it, it will express an sgRNA against ADAM10, which is a tumor suppressor, as I showed in my first part of the talk, and then also sgRNA against P10, which targets the PIK3CA pathway. So if you inject this into the mouse, we would expect a tumor to form, but it would be both green and red. And then we will make a second construct, which will be the same, except that we would have inserted 2 control guides between this SG-ADAM10 Cas9 sgRNA and SG-P10 Cas12, and then test whether this also forms tumor as the same rate as this. And then finally, what this will tell you is that even if you put 2 guide RNAs here for Cas12, if the SG-P10 still works, that means you can still put 2 more guide RNAs here that could be functional. So to finalize that, we would be adding an SG Adam10 Cas9, SG P10 Cas12, and then to shut down the PIK3CA pathway, we'll be putting an sgRNA against PIK3CA and then knocking out GFP. So what would be the final results? From this construct, as I was talking about, we will be getting an yellow tumor, which would be both green and red color, and then it also grows very rapidly. The middle construct would almost should resemble this third construct, but then the first construct will have only red cells and no tumor. Why? Because the GFP, SG-GFP will go and knock out the GFP, which means there won't be any green color. Therefore, only the CRE will turn on only the red color. And then Adam10 and P10 are supposed to form tumors, but then PIK3CA will shut off the P10 pathway, so it won't form tumors in the given time. So right now we cloned all these constructs, and we are trying to make the virus, lentivirus, and once we make it, we'll go into this mouse and do the injection, and then test whether multi-gene knockout is truly possible in the mouse. So in conclusion, I know it's a lot of different stories, but if this would be my take-home message for you, is that we have identified thousands of mutations in most of the cancer types using whole genome sequencing. By using CRISPR methodology in mouse models of cancer, we can identify real drivers among these thousands of mutations, and then also identify what these real drivers are doing. And then we can also take gene profiling data and then apply CRISPR in mouse and identify downstream targets. And if you go in a tumor prawn model, you can even try and test druggable targets. And then for other omic techniques, we need very complex technologies. And the first step, what we are doing in our lab, is trying to develop a 4-gene knockout model in a single cell by combining Cas9 and Cas12. And this could be used not just to knock out individually 4 genes, but we can also do combinatorial screens if we wanted to study in detail. So with that, I would like to thank my lab members, particularly you, Mark, Nahida, and Advay, and then Dr. Nader Sadeghi, who is my department chair, and my postdoc supervisor, Dr. Daniel Shramak, and then several investigators at the MUHC Goodman Cancer Center. I didn't list the name because it'll be a huge list. They are my colleagues, friends, mentors, and I'm really, really grateful and thankful to them for helping me to get started at MUHC for the last 1 year. And thanks to the funding agencies, and most importantly, thank you for all of you for patiently listening to my talk. Thank you.
So thank you very much for such a wonderful presentation. Really nice, excellent work. So any questions, please just unmute your mic and ask. So I was interested in your axis. You showed that there's an axis between Notch, RIP4, IF6, L of 4 axis, and really nicely done. So I also noticed that you had I was wondering why you didn't have NUMB there, simply because it seemed to show that NUMB appears that it can inhibit the Notch signaling.
Yes, that's a good question, Dr. Murali. So NUM is actually upstream of Notch, right? It's a negative regulator of Notch. So NUM would be involved in this pathway, but just upstream of Notch. So what we focused on is after Notch translocates to the nucleus, it turns on the expression of RIPK4, which then goes and phosphorylates IRF6, which then goes and regulates the E11L4 function, right? So we are just looking at that like extreme downstream process.
Okay. So my second question is for the L of 4. which is the last one. So why specifically for that? Because you have other fatty acid elongases. It just my understanding is it's just one of them.
That's a wonderful question. You are talking about one experiment that we are running actually right now. So we made a mouse that carries all the E11L4 all the E11L family mutations like 1, 2, 3, 5, and we are testing exactly the question you are asking. So maybe in the next 5 months we'll have a result which one of this except E11L4 is also responsible for tumor formation.
Okay, wonderful. Thanks you very much. Any questions?
Yeah, I can ask a question. Jean-Jacques Lebrun from the cancer program. Very nice talk. I think it's very impressive to see how you could integrate all those in vivo mouse model with the CRISPR technology. I think it really generates very powerful tools and approaches. And actually, your results already demonstrated that. I had a question regarding the second part of your of your project with the RNA sequencing data. So you are looking at using sequencing data from the RNA-seq. You are looking at the down-regulated genes, and then you generate a mini library of CRISPR knockout, right?
Yeah.
So I was wondering, so that's very interesting because then you can really identify those tumorigenic drivers. But did you look at the other side, like the upregulated genes?
Yes, absolutely. We did that. That's what I said. I didn't have time to show that data.
Okay.
And it's actually active projects in Daniel's lab, my postdoc supervisor's lab. Okay. So we didn't do that in RIPK4, but we did that actually in the notch knockout model because that would be more applicable to lots of patients, right? So what we did is we did an RNA-seq on notch tumors and notch knockout tumors, and then picked up the top 200 upregulated genes, and then went into the notch knockout mouse model and then asked the question, which of the genes are essential to stop the tumor growth, right? And then we picked up a hit, and actually that's basically a very exciting project that's done in Daniel's lab. I mean, I identified already, but then the downstream mechanisms is right now carried out in his lab, and he's keeping that project there. So
okay, could you think of using or generating like a mini library of CRISPR activation, for instance?
Absolutely, yes. That's another work we are trying to do that right now here in here at MUHC.
Okay.
Yeah.
Okay. Very nice talk.
Okay. Thank you, Dr. Lebrun.
Thank you, Dr. Lebrun. Any other questions? Dr. Lapointe?
Yes, first, congratulations, very nice talk. I have a question. Actually, it's more, I would like to hear your thoughts about the prioritization. When you have a list, for example, when you say you did the gene expression and then you kind of rank the different genes, and then you said, okay, I'm going with those. From your experience, did you find that there are better ways than others to rank or prioritize the genes when we do this kind of screening, or like to
That's a good question. But what we did in the beginning was like, I mean, right now you can, if there are other papers where they have looked into different aspects, you can apply those, filters to select the genes. But when we were starting, we were doing this for the first time. So all we did was, okay, we have a cutoff of like infrequent mutations from 10% up to 5%, and then we'll pick up unbiasedly all the genes that fall in between, right? And then that was like about 500 genes. And then the other bottleneck we had is the number of genes we can screen in this technology, right? In vitro, they go genome wide, but in vivo, you cannot, because based on the organ system you're targeting, you have a limited number of cells. So for example, at E9.5, the entire skin stem cell layer is about 150,000 cells, which means if you wanted to avoid double knockouts, you need to infect only 10% of those, which brings you to 15,000 cells. So that's all you have to work with, right? So those are the limitations.
So have you found, so it means that your prioritization is mostly based on the technology that you're going to use downstream to invest.
Yes, and also
in.
the frequency of mutations in patients. So it's between
Okay.
10 and 5%, right? And then anything below 5, we didn't use it. So yeah.
Okay, thank you.
Any other questions? Dr. Philip does excellent work with CRISPR. Dr. Philip, any questions? Any other questions? I think Dr. Philip is not here. Okay, if there are no more questions.
I'll ask one question.
Okay. Hello.
Just this is more like a long view question, because you mentioned that the goal eventually is to develop these technologies for in vivo application, and then obviously you want to translate them to human trials. So just taking the long view approach, can you discuss a little bit on the steps that you need to go through to get this kind of technology to humans and patients? What are the steps we need to take to go from in vitro in a dish to animal models to people?
So yes, so the technology I'm presenting here is mostly for mouse like investigation purposes, right? To identify drug targets or like downstream targets, studying signaling pathways and stuff in mouse. But in terms of applying for humans as a treatment, I think right now there are a few trials going on where you're testing them in kind of transient cells, right? Not in a permanent cells, but kind of a transient cells where you can take them, CRISPR edit them, and then test extensively and put it back into the patients. Those kind of trials are going on, but I think to go into actual human and edit in human is gonna be, I think, decades away, because CRISPR is still not 100% efficient. We are working in the ballpark of 60 to 70, 80% efficiency for knockouts or even activation. And base editing, like to create mutations or correct the mutations, that's even less, maybe around 5 to 10%. But then the off-target effects are there. So it's still like, there is a long way to go within the CRISPR field itself, right? Like here I showed only the application, but people who are working on CRISPR itself, there is a long way to go in terms of like working out all those kinks and the problems.
So if you had to choose a path, what would be a lower hanging fruit? Would it be like blood-borne cancers, leukemias,
something like that? Yeah.
Okay.
Yeah.
Thank you.
Thank you, Derek. Any other questions? So if we don't have any more questions, thank you very much. That was a wonderful presentation.
Thank you so much, Dr. Molly. Thank you, everyone, for attending the talk. Thank you.
Yeah. Thank you, everyone. Have a good week. Bye.